Hello, I'm Ira Jacobson. I'm professor of medicine at NYU Langone Health and NYU Grossman School of Medicine in Manhattan, New York City. I'm delighted to be here with you today to discuss the pathophysiology of chronic hepatitis B, including both the virologic and immunologic foundations of this worldwide infection that involves over 250 million people who have chronic infection. And to our distress, we also continue to see, despite the long-standing availability of hepatitis B vaccine, cases of acute hepatitis B, some of which are quite severe, as we know can happen. I'll start with a personal reflection, which is that the biology and pathophysiology of hepatitis B virus have intrigued me since my medical internship 45 years ago, when I attended a noon conference delivered by a brilliant infectious disease fellow who had come to UCSF from the Brigham, and who later became a very prominent physician scientist in the field. I recall quite vividly being captivated by his discussion about the mysteries of the recently discovered E antigen at that time. We had no idea what it really did. Of course, there have been tremendous advances since then in elucidating the molecular biology underlying the viral life cycle, which is fascinating and much more complex than that of hepatitis C virus and makes the virus far more difficult to cure. Let's review the hepatitis B virus life cycle to provide a framework for our subsequent discussion of the virologic immunologic interactions that are critical to determining the clinical course and outcome of the infection, the ability to clear the virus spontaneously, the outcome of currently available treatments, and the prospects for development of future, far more curative therapies that seem increasingly likely to depend on agents that harness a combination of antiviral and immune modulatory mechanisms of action. Hepatitis B virus infects liver cells through several interactions, the central one of which is the binding of the large hepatitis B surface antigen to the NTCP bile acid receptor on the hepatocyte surface. The mature viral particle is an incompletely double-stranded DNA called relaxed circular DNA, which has one complete strand, the minus strand, and the shorter plus strand. Upon entry to the liver cell and removal of the surface antigen releasing the nuclear capsid containing the genome, the virus releases its incompletely double-stranded genome into the nucleus through nuclear pores, and human polymerases complete the short or plus strand, whereupon the viral genome, through coiling, forms the quintessential covalent closed circular DNA, called cccDNA. The entire life cycle of the virus ensues from cccDNA. Transcription results in the production of 5 messenger RNAs, translation of which results in the production of the DNA polymerase, the core protein and E antigen, which arise from the same region of the genome, and the surface protein in its 3 forms, large, medium, and small. And then finally, the X protein, which has been a mystery for many years, but which has garnered much recent attention for its increasingly convincing contribution to hepatocarcinogenesis. One of the viral messenger RNAs serves as pregenomic RNA, the template for a reverse transcriptase function, much like HIV, subserved by the DNA polymerase, and resulting in the production of the new long strand of DNA, then the production of the short strand of DNA. It should not escape notice that the DNA polymerase of the virus also has a reverse transcriptase function, as I mentioned, making one think of retroviruses. And it may not be a coincidence that some of the same antivirals, most notably tenofovir, though not entecavir, confer profound suppression of viral replication. Going back to the HBV replication process, this can only occur if the genomic material is already enveloped inside a preformed intact nucleic capsid produced by the polymerization of multiple core protein molecules. The resultant particle, the nucleic capsid, is then enveloped by the hepatitis B surface antigen in the Golgi complex, and the final virion is either extruded from the liver cell into the blood or releases its relaxed circular DNA within the cytoplasm of the same cell in which this life cycle has been propagating to initiate the life cycle all over again. This accounts for how cccDNA continues to be replenished in patients with chronic infection. Of critical importance, a small proportion of the new viral particles contain a double-stranded linear rather than circular DNA, such as I've described, and this is produced by an alteration in the replication process, and it is this and only this form of DNA that integrates into the host genome. The portion of the genome that integrates is limited, however, to the sequence from which hepatitis B surface antigen can be produced, and, as suggested in recent literature, the X protein as well. The integrated DNA is widely thought to contribute to oncogenesis by causing mutations, either in the form of chromosomal translocations or insertion of viral genetic sequences at critical genomic sites, such as proximity to tumor promoter or suppressor genes. HPV-associated liver cancers, or hepatocellular carcinomas, contain numerous cells with integrations, and there is now little doubt that integrated viral DNA is responsible for non-cirrhotic HCCs in HPV-infected people, even though most other liver diseases that lead to HCC mostly do so in the setting of cirrhosis. Doubtless, integration plays a role in cirrhotic HPV patients as well in inducing liver cancer. There are also several biological effects of the X protein that scientists believe contribute to oncogenicity. Turning to the topic of the role of the immune system in the pathogenesis of HPV-related liver disease, an essential point is that the virus is not considered to be directly cytopathic to hepatocytes. Rather, the disease we call chronic hepatitis B is mediated by the immune response to the virus. At the time of initial infection, there is a diminished innate immune response featuring blunted production of type 1 interferon, resulting in widespread infection of hepatocytes. In infancy, perinatal transmission results in chronic hepatitis B in over 90% of cases, associated with a high level of regulatory T-cell activity and an immature CD8 T-cell response, amongst other potential factors. With maturation of the immune system, the rate of chronicity after acute infection falls dramatically as childhood progresses, and in adults, it's about 2 to 5%. Control of acute infection ultimately depends upon a strong CD8 cytotoxic T-cell response. Viral antigen and presentation by CD4 plus lymphocytes are essential for supporting the CD8 response needed for viral clearance. In fact, studies in which experimental models are depleted of CD4 cells show that a strong CD8 response cannot be mounted without intact CD4 helper cells. Natural killer or natural killer T cells play a role as well, particularly through interferon gamma production. So you can see how many different parts of the immune system are involved in the pathogenesis of hepatitis B, even in the acute stage. Despite the initial weak interferon production by innate immune cells early in infection, innate immune activity is involved in the recruitment of adaptive immune cells. Recently, increasing attention is being given to the role of B cells as well, with emerging data focusing on the role humoral immunity has in viral clearance. The immunopathology of chronic hepatitis B is incredibly complex, and virtually every aspect of the immune system has been evaluated and published upon for their contributory roles. Early in the course of chronic infection, hepatitis B-E antigen acts as an immune tolerogen, which downregulates the CD8 T-cell response. This is associated with what's previously been called the immune tolerant state, now renamed chronic HBe antigen positive infection, characterized by sometimes astronomical HPV DNA levels that can exceed 100 million or even a billion international units per milliliter of serum HPV DNA, yet little or no inflammatory activity in the liver. It gets more complicated, though, because recent studies, for example, led by Patrick Kennedy in the UK and others, have dispelled the notion that there's no T-cell activity at this stage. And again, that's why many experts have gone away from the term immune tolerant in favor of chronic HBe positive infection. An essential immunologic element as chronic hepatitis B evolves is the phenomenon of T-cell exhaustion related to the expression of high levels of viral antigens. In vitro studies of T-cells from such patients demonstrate impaired expression of markers of cellular immunity which perpetuate chronic infection. Perhaps most strikingly, CD8 T-cells exhibit high levels of inhibitory molecules that are intrinsic to the perpetuation of the chronic infection state. These include molecules such as PD1, CTLA4, IL-10, and TGF-beta. PD1, expressed on lymphocytes, and his counterpart in the liver, PDL1, have garnered the most attention. PDL1 is expressed on tumor cells in certain organs, including the liver, and PD1 binding to PDL1 inhibits immune function, such that immune function can be restored by either PD1 or PDL1 inhibitors, both of which have been or are being studied in hepatitis B. This accounts for why checkpoint inhibitors, as well as PDL1 inhibitors, have been found to be capable of restoring immune function, and in the process eliciting hepatitis flares by restoring immune function, and in very exceptional cases when used as an investigational therapy, the therapeutic goal of actual HBsAg clearance. However, this drug class is also associated, as you know, with significant potential for toxicity, especially to the thyroid in our hepatitis B studies, but more seriously, including the liver, again, resulting in the potential for significant hepatitis flares. This class of drugs remains part of the panorama of immunologic agents now being studied. Other classes of potential immunotherapies include therapeutic vaccines, monoclonal antibodies, toll-like receptor agonists, and T-cell receptor therapy. I've alluded to the humoral immune system briefly before. There's emerging literature that has added to our understanding of the role of B cells in chronic hepatitis. For our purposes here, the role of B cells is underscored by the phenomenon of reactivation of hepatitis B, even in patients with core antibody alone, as a marker of resolved infection after exposure to certain agents that inhibit B cell function. Indeed, these patients may still harbor cccDNA as well as integrated DNA, but let's remember that it's only the cccDNA that can lead to reactivation. I'm sure many of our listeners are aware of the risks of reactivation and other CD20 therapies. These drugs deplete circulating naive and memory B cells, and the ensuing reactivations that can occur, and sometimes be quite severe, emphasize the importance of the humoral immune system in control of infection, and also reminds us of the need to cover these patients with antiviral therapy with entecavir or tenofovir before they begin such therapy, and indeed well after, at least a year well after, the last exposure to these drugs. Several excellent reviews of the role of humoral immunity in control of HBV have been published recently and are easily found in PubMed if you'd like to read further about this. The importance of the immune response to chronic hepatitis B is illustrated by findings in ongoing therapeutic development programs. Even the use of agents classified as antiviral drugs involves to variable degrees the potential for such drugs to elicit an immune response that may be key to delivering functional cure. The central concept is that agents that suppress viral antigen expression, especially of hepatitis B surface antigen, may in effect release a hitherto suppressed immune response brought about by chronic presentation of large amounts of viral antigen. The technical definition of functional cure in current clinical trials is clearance of HBsAg, or hepatitis B surface antigen, 24 weeks after cessation of any therapeutic regimen, including the traditional nucleotide or nucleoside agents, namely tenofovir and entecavir in the current era. Studies of several small interfering RNAs that bind to targeted sequences in the messenger RNA for HBsAg and inhibit its formation have demonstrated marked declines in HBsAg levels, with slow rebounds after discontinuation and rare functional cures. Similar findings were noted with other siRNAs, and it's become clear that siRNAs will not suffice as monotherapy and must be combined with other agents, whether antiviral or immunologic. Accordingly, siRNAs have recently been studied in combination with interferon, which has potent immunologic properties. Interferon has outperformed other immunomodulatory therapies, particularly TLR agonists studied thus far in combination with siRNAs, although there is much more work to be done to address this question. But interferon is back in the investigative arena in hepatitis B. The general efficacy of the antisense oligonucleotide class compared to siRNAs as monotherapy is generally attributed to toll-like receptor stimulation of innate immune pathways that occurs after exposure to antisense oligonucleotides, and that's in addition to its antiviral effect of HBsAg knockdown. This is also manifested by the occurrence of ALT flares, which are immune-mediated more commonly with antisense oligonucleotides so far in studies than seen with siRNAs. A final illustration of the interest in harnessing the immune response to cure hepatitis B is the range of other immune modulatory therapies being tried. These include therapeutic vaccines, such as viral vectors, that express various combinations of surface, core, and polymerase proteins to elicit an immune response, monoclonal antibodies to HBsAg, which not only reduce surface antigen dramatically as what could be called an antiviral effect, also form immune complexes which may elicit a separate immunologic effect. And finally, T-cell receptor therapies, which upgrade the immune response to surface antigen expressed on liver cells or potentially liver tumor cells. It's hoped that antivirals, likely combined with one or another immune-based therapy, will enable us to help the over-chronically infected people around the world. It's an exciting time. We're still climbing up the mountain, but we hope to reach the peak in the next few years. And I thank you for listening.